anyway, so we'll start with a quick intro about medicinal genomics and what we do, and then we'll jump into the technology that I'm going to be talking about today, which is called youPCR. We'll go into a little bit more detail about how youPCR works and the mechanics of it, and then we'll jump into the different applications of it. So one of them being gender testing. The other would be determining different types of cannabis plants, so types 1 through 4. And then also how these tests can be used to screen for different plant pathogens and breed resistant strains. And then we'll dive in a little bit about sampling techniques and things like that. And we'll also talk about kind of the next step to this, which is using quantitative PCR and how that can also be beneficial. So what we have that we're talking about today is simple presence absence, but when you get quantitative, there's a bit more that you can do with that type of data. So with that, I'll dive right in. So a little bit about medicinal genomics. We're a leader in cannabis safety testing. We're really pioneering the way in microbial testing for cannabis, especially on the molecular side of things. We also are a leader in cannabis genetics, so we put out one of the best reference genomes in the world to date. So now we have a fairly complete cannabis genome. We've got it all assembled into nice chromosomes and everything, and it's a bit bigger than we thought. So there's a lot of discovery to be made there. We're now busy annotating that genome and figuring out what it all does. And then, of course, we've got our point-of-grow genetic testing solutions that I'm going to talk about today. So yeah, all the different solutions we offer here, mainly going to be focusing on that early detection platform for plant screening. But of course, we've got DNA purification assays that are specific to cannabis and different cannabis products. So one for flower and one for infused products and extracts. Those are all used in conjunction with microbial testing. But then we also do sequencing as well. So of course, having the best reference genome in the world allows us to actually get a lot better quality data about what cultivar you're working with and also identifying specific traits that might be of interest agronomically and chemically. So the workflow of this platform looks something like this. So it starts off with your leaf sample, or sometimes if we're doing certain types of plant pathogen tests, we'll go with stem. But basically we'll take a biopsy punch of that, 4 millimeters, and then place it into a solution, which is basically a lysis. So this breaks open those cells so you can get the DNA out. And you're going to boil that for 50 minutes. That's what the action that's actually getting the DNA out. Next, we're going to take that DNA, a little bit of it, and add a couple more reagents. So that, you know, if you're, how many of you are familiar with PCR in the room? Okay, so most of you actually, great. So yeah, you know, master mix, primers, things like that. Your purified DNA is going to go into that well, and then we're going to close it up for another 35 minutes, and it's going to do its cycling, and then we see the results. So pink would be a negative for the gene of interest that we're looking for, while a yellow would indicate that you had the gene of interest present. So I don't want to bore you guys too much with this because it sounds like you know, but for those who don't, how PCR works is we basically take that purified DNA, and then we add in a few other things. So primers that are specific to our target sequence, as well as DNTPs, which are dinucleotide trisophosphates, the things that are basically going to make your new strands of DNA as you're doing the amplification. And then we've got our polymerase, which is actually doing the work in order to make that new DNA strand. So a visual of this, we've got our double-stranded target, right? So we're going to heat that to 95°C. That's going to separate it out now into 2 single strands. And then when we cool this down, now we've got the primers in there with all the other components. Those primers are going to anneal to their complementary sequences. And those are upstream and downstream of the actual target gene sequence. And then once we heat this up again, now we've got elongation that's happening and extension. And we're actually creating new strands of DNA. And that's a single cycle of PCR. We're going to do this over and over again to the point where we get many copies of this thing. Now, the only thing that's changed slightly with youPCR is we've now added into this mix a pH-sensitive dye. And that pH-sensitive dye is going to be what's going to allow us to get that colorimetric answer of pink and yellow. So every time we get a round of amplification in this assay, we're kicking out a proton. So if you remember your general chemistry, kick off a proton, you're getting more acidic. Every time you get more acidic, you're going to get a color change. So that means every time you're getting that amplification event and showing that that gene is present, then you're going to slowly but surely shift over from pink to yellow. And what's great about this platform is it can really be done on very affordable instruments. I actually brought one here today for a little show and tell. So this is a mini PCR. It's got 8 wells. It also comes in a 16 well format as well. And this thing is super inexpensive. This is $650. When you're thinking molecular biology, that's like chump change, right? And then if you wanted to expand a little bit and do higher throughput, you could also get a 16 well version for $850. What is great about these things is, of course, as you saw with the small size, they're field portable. So you could take these things out to different cultivation sites and get an answer very quickly. That whole process takes about 2 hours start to finish. And then for those of you who are doing more large-scale cultivation, you can get the standard thermal cycler, which is usually 96 wells, but some of these even come in 384 well formats, and these things are relatively inexpensive, about 3,000. So not quite as cheap as the Mini, but still, when you're talking molecular biology, this is really not over a high barrier to entry, right? So I'll jump in now to some of the different applications. So the first being gender testing. So no one likes males, right? So want to get rid of those as soon as possible. And the benefits of doing this gender screening in-house is it's fast, right? So you can do this in under 2 hours. You can do it en masse if you need to. It's accurate. It's greater than 98%, or it's about 98% accurate. The reason why there's a difference of 2% there is because we can't rule out hermaphroditism, right? So hermaphrodites are actually genetically female. So they don't have Y chromosome, which is what we're targeting in our gender testing assays. However, if you stress out a plant to a point, right, it's going to start freaking out. It sometimes can start to express these pollen sacs, which then cause a lot of problems for your cultivation because now you have seeded females. So that's the 2%. But again, if you know your cut pretty well, you should be able to mitigate that potential risk. And then furthermore, this whole thing is compliant, right? So of course, we're still dealing with a Schedule I drug that's federally illegal. So if you're going to ship anything with any appreciable amount of THC, especially over 0.3% THC, you could have the DEA come and knock on your door. So always good to be able to not have to send this thing off to somewhere outside of state and just get an answer very rapidly. And so we've done some validation work with this to demonstrate. This is just some of the early stuff. We've done quite a bit more, and this product's been out in the market for a while now. So everyone's been using this without any issues, but this is something we did with Colorado Seed, and we basically compared this to our quantitative PCR version of the assay. So sounds like a lot of you are familiar with PCR, but quantitative PCR, the only difference here now is we're adding a fluorescent probe, and that instrument is now reading the fluorescence that's coming off every time there's that round of amplification occurring. One benefit of going with the more expensive instrument, which is the qPCR, is you get also an extra resolution of data. So you can get, basically we target cannabis DNA for the internal control that is not Y chromosome. So, and this is, again, quantitative, so those are expressed in the CT values there. But basically this serves as a nice double check to show that your DNA extraction was indeed complete, because otherwise if you're getting just a simple yes-no answer, what if you didn't extract the DNA, right? And now suddenly it's a negative, but there's no DNA in there. So this is serving as a nice double check. So we can be very confident 100% in these answers. But as you can see, 100% concordance out of the 91 samples tested. So it's pretty dead on, just don't hermy it up, right? And then of course, when it comes to time, a lot of people for some time were sending these things off to different services. I won't name any names, but ultimately those services weren't being very honest with what they were doing. And also it kind of stirred up a lot of controversy in the community. So this is a great way to be able to keep your genetics in-house while not having to send it out. But basically, yeah, you can get this all done, and you can get up to 470 plants done in a day if you're filling these 96 well plates in under an hour. I don't know why I have my total time missing, but basically it's about 2 hours. If you're filling the plates, right, it takes a little extra time, but roughly the actual running of the process takes about 2 hours. And we've also got these reagents, at least for the lysis, pre-aliquoted, so it's very user-friendly. We have these little disposable grommets. We were previously using biopsy punches, but we really like to think that we stoner-proof this thing, because before, we were using these biopsy punches, we're like, oh, this can't be that hard, just sterilize it, right? But of course, no. Most cannabis cultivators are not really too familiar with pipetting and doing molecular bio work, so we ended up moving to these disposable grommets. The cross-contamination rates have pretty much plummeted, so it's a lot better of better way to do this. It's a better system. But basically, yeah, you do your hole punch, take a toothpick, or if you're a lab guy, pipette tip, place it in the lysis solution, and you're ready to rock. So the next application of this would be plant type determination. So how many of you are familiar with the name Etienne de Meyer? That's, yeah, I'm not too surprised, this random researcher guy, but basically he classified all the different types of cannabis that are out there in terms of their cannabinoid content. So that's the type 1 through type 4s. And that's exemplified here in this figure by Ethan Russo. So we've got, I want you to focus your attention here on those arrows. So we've got cannabigerolic acid there, and that is the precursor for all of your major cannabinoids that you find. We've got minors like varins and things like that, but we're going to focus in on that. And so the two primary pathways that you're going to be pushed towards are either THCA or CBDA synthase. There's also CBCA synthase, right? But CBCA is something you find mostly in hemp varieties, and it's never really a dominant cannabinoid that we see. It's mostly in trace amounts. So we're going to zero in on those two synthases in particular. But essentially, with these type 1 through 4s, you've got different qualities. So type 1 would be THCA dominant, producing very little CBD, basically similar to exactly what you find in the vast majority of dispensaries today, your drug-type cannabis. And then you've got type 2s, which produce both, so THCA and CBDA. Then we've got the type 3s, which are CBDA dominant, so very little THC. This is kind of like what you would consider a hemp cultivar, right? Something below 0.3% THC. And then we've got type 4s, which are CBGA dominant. So if we rule out the CBCA synthase portion of this, and you have no THCA or CBDA synthase present, then you have only one place to go. There's nowhere for the pathway to continue, so you get a CBGA dominant plant. And this is what it looks like on youPCR. So type 1, right, we're positive for THCA synthase and negative for CBDA synthase. For type 2, we can produce both. We have both enzymes. Type 3s, we've got are positive for CBDA synthase, but negative for THCA, similar to what you would see in a hemp. And then type 4, you'd be negative for both, so you'd produce naturally mostly CBGA. And then we can further segment these different plant types and look at all the different alleles, right? So you could be homozygous for that THCA allele, or you could be heterozygous for it in a 1B. And then similarly, the same idea here for type 2s and 3s, basically just looking at what types of allele frequencies you get. And we can further elucidate this stuff in much greater detail with things like sequencing. Unfortunately, that little instrument cannot do all of this, but if you're willing to shell out a little more, we can find out a lot more genetic detail about what this can do for you. And this is actually, this technology was something that a company out of Oregon called Oregon CBD did, and they used our techniques along with HPLC in order to figure out if their breeding projects were going in the right direction. So these folks ended up actually breeding a pure CBGA line, and they did that using our tests. So they were able to figure out, all right, this guy has a CBDA synthase, we don't want to include that in the breeding project, that kind of thing, and stabilizing over and over many generations. One thing I do want to caution, though, is just because you don't have a certain cannabinoid synthase, that does not mean that you cannot produce that compound. So let's take the example of hemp, right? So you've got CBDA allele present, you're positive for CBDA, but you have no THCA synthase. Just because that THCA synthase is not there does not mean you're always going to be below that 0.3% threshold for THC to define something as hemp. And the reason for that is these cannabinoid synthase enzymes, there's many of them scattered throughout the genome. We don't actually know what some of them do. There's 18 full-length copies of THCA synthase, and only one of them that we know of is actually active. And then there's 30 THCA-like synthases that are out there. We don't know what the heck all those 30 do, but they're there. And but basically, the enzymes that are in there that are cannabinoid synthase related are all promiscuous. So you could potentially get more than 0.3% THC, even if the THCA synthase is not there. So again, it's something where you still need to do confirmatory HPLC testing. There is no substitute for that, but this can certainly give you early clues as far as how to drive your breeding projects forward so that you're not wasting time on crops that won't actually lead you to the answer you want. The next application of this would be pathogen detection. So the benefits of doing this pathogen detection are a few. So firstly, let's give the scenario of, I don't know, John down the street has wedding cake. And you're like, man, I really need to get that cut of wedding cake because it's got everyone super lit and they're so happy. So I want to get this, this wedding cake, but I want to make sure that John's doing honest business with me. I don't really trust John's growing techniques, and I've seen PM on his plants before. So if I'm going to bring in this wedding cake into my grow room, I want to make sure that it doesn't have PM on it. So this can be used on site. You can go down there with your mini PCR and say, hey, John, I'll let you know in about 2 hours if I actually want to do business with you. So if you turn out, you might not want to stop by and get that wedding cake, maybe go somewhere else. And then furthermore, you could also check your moms and make sure that, say you're cloning out 100 different plants, or taking 100 different cuttings. Maybe you want to look at those different sites, maybe 10 different sites, right? And make sure that you can test clean on all 10 different sites so that you can at least get some accuracy, right? In terms of knowing that you're going to produce clean clones. And then also just in general, it's good to periodically screen your grow room. So if you're someone like John who's very careless and lets random people into your grow room and they've been touring other grows, you probably don't want them coming into your grow and potentially contaminating it with whatever plant aids they have, right? So again, just another application here, and I'm going to go into a little more detail about all the different assays we've developed. So the first being powdery mildew. So powdery mildew is it's a really tricky one. So here I've got a figure from a study that looked at powdery mildew in hops. And so DPI stands for days post-inoculation. And you'll notice after a few days, 4, 5, 6, you start to get these mycelial networks that start to form around the plant. And so this is something that you can't actually see with the naked eye. This is just kind of an invisible cloak that starts to envelop the plant. And you're not going to be able to pick it up. People can visually see PM. It's already too late. It started to sporulate, and you have a problem. So what's interesting, especially about PM, though, is PMs are very unique, and they've evolved to their particular hosts. So you've got hops, which is infected by something called P. macularis, and then you've got Golovinomyces, which affects grapes. So we actually, when we were designing these tests, we took the primers from the literature for each of these, and we tried to amplify it on known cannabis PM, and we're like, we're not getting any hits. Nothing's happening here. So that led us next to be like, well, this doesn't work. Let's figure out what the heck it is. So we sequenced the powdery mildew genome that infects cannabis. And I don't know if any of you have seen me speak on microbial testing, but I talk about these things called internally transcribed spacer regions, or ITS regions. These things are like molecular barcodes, so that they're basically a way to pinpoint exactly what type of organism you're looking at because they're evolutionarily conserved, but there is a lot of variability in them, which gives the nuances to what species you're looking at. So it's 98% identical with P. macularis and Golovinomyces, whatever this cannabis-derived powdery mildew is. So it's one of these things where you have to make sure that you're actually hitting the right target, right? So we figured out we have a novel species here in cannabis. And the other thing, too, is if you want to pick this stuff up and you think you're going to be able to do this with what a lot of testing labs are doing, California excluded because they're all molecular, but a lot of people will plate stuff thinking that they'll be able to see some of this harmful stuff. But powdery mildew doesn't really culture. So if you think you're going to pick up your PM doing that, good luck. So molecular is the only way to go with that. So until someone tells us otherwise and that we're wrong, we've affectionately called this thing Golocanomyces. And basically, with the same idea, right? We're going to do a hole punch, and we're going to boil it, take a little bit of that DNA, 2 microliters in this case, and then we're going to add all our reagents and get a colorimetric answer. So here are just the different examples of that. And one thing I do want to note here about our tests is we actually don't pick up the spores, and that's actually a good thing. You might wonder why would that be a good thing? If I can't visibly pick up PM when it's visible, what's the point? Well, the point is to pick it up before you have the visible sign, right? And then furthermore, we have a lot of these spores that are just hanging out in the environment, like single spores. You don't want a single spore that is dormant to actually trigger a positive, because then you're like, well, I have a problem. But it does lyse the mycelium, those mycelial networks I was showing you in the earlier slide. And so that's what's allowing us to detect specifically the early stages of powdery mildew, rather than before it's when it's too late and now you've got your whole grow room covered in PM. And also, to add to that, if you know that you have a plant that's testing positive for the mycelium, then you know that plant is now susceptible to PM. Now, if you're getting negatives across the board always with your plant, then that might be a good candidate, right, for your breeding project for a PM-resistant plant. So. And this can be applied, same idea can be applied to russet mites and things like that. These are obviously bugs, so not quite the same. But the same idea applies when it comes to why do we design these tests. We design these tests to make sure things that we can't normally see easily with the naked eye, now we can see them. So we set after doing this with the russet mites, so we sequenced the russet mite genome. The problem with these things is there's an explosive amount of growth with these things. They lay eggs very quick, 10 to 50 eggs per adult. So they can get out of hand very quickly. And the fact that these eggs are just kind of like hidden in the petioles, leaves, and stems in very tricky places, and they're not really easy to see with the naked eye. When you take a hole punch of this stuff and can get really in depth, now you know for sure if you got russet mites or not. And so here's just an example of clean versus mited out leaves. We always want to run controls with these, right? So on the right-hand side, towards the bottom, you've got pinks, which are your no template controls, no DNA added, and then you've got a positive control, which is the bright yellow there, showing that that the test is working right and we're picking up what we're supposed to. We've also done the same thing with Botrytis cinerea. So what's really important, this is bud rot, right? ever broken open a nug and then saw a bunch of fuzzies, that's bud rot. So what's important about this too is being able to get very, very sensitive specificity with this, because once you have a little bit of botrytis, it can get out of hand very quickly. So here's a series of dilutions. You can see at the top, we can get down to a 10 million fold dilution, and we can still pick this stuff up. Which is going to be very helpful in the case of this, maybe not so helpful in the case of something like a PM spore, which again, we're not lysing, so we're not getting DNA. But either way, just another application of this. And the thing is with botrytis is you're not always going to be able to see it very easily. So if you look at this positive here, you might be able to look, if you look closely on the magnifying, or the scope there, you might see a little bit of powder, or a little fuzz, but you're not really gonna get, you're not gonna really see this with the naked eye very well, but it is positive. And then the negative, again, you can't really see much of anything, which you shouldn't, but they look virtually indistinguishable, ultimately. So just tools to make sure that you're picking things up that the eyes can't see. The next application that we've developed this for is fusarium. This is actually, I should have updated this slide, but we are targeting now Solani as well, rather than just oxysporum, because that has come to our attention that Solani is something that we should be looking for, and is also something that's a pain for cultivators. So we have added that primer set into our assay. But basically, what fusarium does is an infection that starts in the soil, travels up through the roots into the xylem, and if you were to make an analogy of this, it's basically like take a human and you clog its veins, and now suddenly you can't get the nutrients, you can't get the blood to where it's supposed to go. In the case of the plant, right, it's xylem and phloem. So you can't get the nutrients to where it needs, and therefore your plant starts to suffer and die. And then you get things like dudding, where you see a perfectly healthy plant, and then one branch is just like, didn't quite make it, right? So we've developed tests for these, and we've got both the regular youPCR version, colorimetric, simply yes, no. We've also got the quantitative versions as well. And so why would you want to do qPCR to quantify pathogens? Well, this way you can actually see how infected you are. So here's some data that I did when I went up to Moss Landing and did some field testing. And I sampled different areas of the plant with known to be infected with PM. And you can see that it's reliably detected throughout the plant. And when you can quantitate, you're not so reliant on the simple yes, no. Because so these CQ values, basically, anything above 35 would probably not be flipped on youPCR, but it would on qPCR. So if you spend the extra bucks, you can get the extra resolution. I did the same thing here with fusarium. The data pretty much supports the same idea as what I kind of just described. Since it's coming from the soil and coming up through the roots and into upwards in the plant, it's probably going to be easier to sample this, right, and get it at the bottom of the plant rather than the top, and the data reflects that is mostly true. Now, one thing about fusarium that is tricky, right, because it does start at the bottom and goes through the stem and upwards through the plant, you're going to detect it still, as you can see in the leaves to some extent, but it's not as easy to detect in the leaves as it is with just getting a stem core, and especially if you go lower and lower on the plant, because that's going to be a better chance of picking it up. But it is detectable throughout the plant. But if you're going with something like a youPCR solution where it's a simple yes, no, try and get as much of the concentration as possible lower in the plant, right? And then furthermore, it's a great tool to be able to actually monitor the progress of how you're trying to remediate this stuff. So before would be, filthy, completely ridden with PM or whatever plant pathogen, and then you can monitor that treatment program and see, did I actually get a reduction in DNA of the pathogen, so I know if my treatment is working or not. So that's all I really had for you guys today, but yeah, I'd like to open the floor to questions.
Anybody have any questions for Kyle?
Most likely that will be the case. Starting to bring in viruses as part of your testing protocols.
Viruses are a little trickier, and also I think there's a lot of viruses that infect cannabis that we haven't yet identified. Actually, so there was a panelist earlier that I was speaking with, Daniel Grace over at Dark Heart Nursery. They've recently rolled out a new test to test for something called hop latent viroid that actually a lot of people thought was fusarium for a very long time because it presented the exact same dudding symptoms that you would see in fusarium. However, this is a viral infection. These things never pop for fusarium. You've got things like tobacco mosaic virus. I know there are companies like Agdia that have immunoassays and immunostrips for them. But at this time, we're not really pursuing the viruses. We're mostly diving into the cannabis genome and annotating and figuring it out and what it all means.
I've got one back here. So somebody gave a presentation in which they indicated that specimens with powdery mildew were all negative for other potentially more dangerous infections. I know people don't want to smoke cannabis with powdery mildew on it, but is there a potential for a, an intentional mycological relationship which doesn't take away from the user experience that could fight, more malicious infections?
I think there is a better way of doing that than inoculating your plant with PM, right? I mean, yes, it's an interesting trend that colonizes, and then suddenly we don't have things like Aspergillus or Morphusarium or other things that might be not so great for human health. But you can also use things like Bacillus, good probiotic that will prevent the colonization of some of these things. Also, Trichoderma is another one that people use. That one's actually known to displace and eat the Aspergillus and Penicillium and other types of things that aren't so good. What was that? Exactly.